Okay, welcome back. I will talk about aggression and 5-HT, and I think most of you have probably encountered this situation, and maybe you've been the victim of such a person, or maybe you even turn out to be that person yourself. So we all have, of course, this tendency that some certain things can make us aggressive, but the spectrum of aggressiveness is quite different. And serotonin has been quite profoundly implicated in aggression. So it's known from animal studies and from cerebrospinal fluid measurements from violent offenders that low serotonin levels can actually elicit aggression. So we set out to study this a little. I'm going to talk about a project that was just finalized, but what the question is, of course, what receptor mediates aggression? There is some animal literature to supporting that 5-HT receptor knockout animals are more aggressive, and if you stimulate the 5-HT1B receptors with selective agonists, you get very placid and well-behaved rodents. So the 5-HT1B receptor seemed to be an obvious target for the effects of aggression. And the 5-HT1B receptor is a quite complex receptor in that it has both a function as a presynaptic autoreceptor, where it sits here and constantly monitors the 5-HT levels in the projection areas. So not so much in the raphe. You also see a little 5-HT1B in the raphe, but it's mostly in the presynaptic function. But it's also located as a heteroreceptor, as you can see here. So it is a complicated receptor, and if I have time, I'll get back to this complexity and how you can interpret outcomes in different ways. So the questions that you can ask yourself is, are the neural circuits of aggression dysfunctional in individuals with extreme aggression? And how do you find extremely aggressive people? Well, you go to the most secure prisons you can find, and you try to convince the probation and prison service that you can actually take these very dangerous people and bring them to a scanner. And that takes a little time to convince them. And I can tell you, we actually also had some wild plans about doing acute tryptophan depletion. But at that point, the forensic psychiatrist told us that he thought that would be better if we did that in the prison and not outside the prison. So that part we had to skip. But after a long and intensive negotiations, also with the ethics committee, we were actually able to get permission to transport these very violent offenders, murderers, rapers, and so on, from very, very secure prisons. A very interesting experience, I'd say. So they had an armed guard with them while they were being scanned, and fortunately, I can say nothing happened. They came back, and they didn't run away, and they didn't...
Did you use the armed guard as control?
No, I could have done that. I have a picture of the armed guard. Well, anyway, so is our serotonin 1B receptors involved in human aggression was the next question we were asking. Then are these neural circuits of aggression influenced by serotonin signaling? So these were questions that we were asking, and we had 3 different columns of measures, one being the molecular with PET measuring serotonin 1B receptors, another one measuring brain function with functional MRI in an aggression-related paradigm. And then finally, we had measures of the trait, anger. So these would be more personality measures, self-questionnaires about these people. So by combining these 3 different items, we were interested to see how we could link these different traits together to the neurobiology. And here we have, of course, we also had some more non-aggressive individuals as controls, and these are the neural correlates of aggression during an aggression-inducing paradigm, and the PET measures of 5-HT1B receptors, and we had the questionnaires as well. So you may want to think of it as a group-based analysis, because we have, on one part, we have these highly impulsive, aggressive offenders, some of them being actually also psychopaths. And then you have some more low aggression people. So you could think about it as a group-based analysis. On the other hand, you could also think of it as more of a spectrum. So, so you will actually see that the traits that these people had would be not quite as distinct as one would have thought. Maybe because being a violent offender in a prison also tells you something about socioeconomic and cultural traits. So if you are brought up in a very non-violent environment where it's not culturally acceptable to conduct violent acts, then you are most likely not going to end in the prison and vice versa. So it turned out to be quite a spectrum. And what we often see is that we have this tendency that we want to look at group differences, but in fact we are looking more at a spectrum of different things, and it often turns out to be more informative to look at the different spectrum in relation to your neurobiological measures. So that's what we did, and the neural circuits of aggression. So one quite obvious thing to look at would be how the prefrontal cortex can exert an inhibitory control over amygdala and striatum that are obvious candidate regions for aggression. So how do you make people angry in a scanner? So that's a question here.
You know, the histamine also, initially serotonin and histamine stored in the same compartments. So do you think sort of the pharmacological manipulation somehow also change the histamine, and then you measure to the signal is resulted by histamine changes in addition to serotonin?
So the question is whether we looked at histamine, and the proposal is whether one would...
Not directly the histamine, but maybe it may somehow also change the serotonin activation levels.
Yeah, we did not look at that. So there was another question here.
Yeah, so do they get paid, or do they just volunteer because they want to get out for a day?
Oh, you mean the aggressive individuals? Well, if you think about it, there's not much to do in a prison. You know, you have to pass through 2 big walls, and you have to leave your mobile unit, and it's very, very secure. What do you do in such a prison? You can't really do much. You can work, and you can earn a little from working, and then they are also supposed to make their own food and clean and things like that. But it's pretty boring, I'd say. And we had no intentions of recruiting only males, but it tended just to come out like that because the prevalence of violent offenders amongst males is so much higher than among females. So we didn't recruit any females for our project. We had a long discussion with the ethics committee, and we ended up actually paying them the same amount that the healthy volunteers received in order to participate in the study. And I think we were very fortunate. First of all, I had 2 very bright and good-looking female PhD students, a psychologist and an MD, that went out to the prison, and everybody was very eager to show off, of course. And as you know, in a prison you often have a hierarchy, so there is one who is the alpha male, so to speak, and everybody has to obey to his rules, and just coincidentally he happened to be the first participant, so he could come back and brag, and everybody wanted so badly to join. So there was one particular very, very dangerous murderer who, including murdering his own brother and children and terrible things. He very badly wanted to take part in this project as well. He was not allowed to leave the prison, but he could have joined the neuropsychological investigation. So we were able to do, in a much larger sample, the neuropsych investigation. but we opted out because his plan, he's very smart, his plan was to, I'm sure, to use the neuropsych data to kind of convince the court that he should have a less of a sentence. So this is a very interesting discussion about how can you be excused if this is what my brain looks like, you know, I'm excused. So that's how they think about it. There's a question down here. No?
No, just I had some question. I was just wondering about the feeling of restraint.
Yeah, in the scanner.
Yeah.
Feeling restrained in the scanner, does it provoke anxiety, for instance?
Kind of stress,
That's-
anxiety.
Oh, that's a wonderful question. I really think that's a very, very appropriate question to ask. So do people feel anxious when this? How many in this room have tried to be in an MR scanner? So quite a number of you have been there, and to me it's a tight experience because you get, you know, you have the coils very close to your face, and if you move your arms a little, you can't really stretch out. So it is a bit claustrophobic, and I think that it might be a good idea if you have the opportunity to actually have a mock-up scanner so you can bring people into the scanner so they know about the noise and how tight it is and all that. Because I think there might actually be an interesting point in how do people react the first time versus the next time. And we've actually looked at test-retest with functional MRI, and there are some papers out there, and the initial studies suggested that there was a quite good reproducibility when you take the same subject and expose them to the same paradigm. I'd say that I was surprised to see how badly it reproduces. So I would think that doing the same subject twice would be way better than doing it, you know, in different people, but that's not the case. Yeah.
Yeah, and then also how this could correlate with the aggression measurement, like for the
Yeah, I mean,
structures.
so I mean, in our case, we really wanted them to get angry in the scanner, because that was the whole point about it, that we, you know, the more angry they would get, the better we could look at the responses to, and not only their aggressive behavior, but also we could correlate the behavior to the hemodynamic response that we were seeing in the whole group. So now the problem was that at that time there were some established aggression paradigms, but they were not made for scanning. So it's something you do outside the scanner, and then you could. So we had actually to come up with a novel fMRI paradigm first. Of course, we would have the self-reported personality tests that we could use at different scales, and we took a number of different scales just to make sure that we didn't miss out on anything. Then we did some laboratory measures of aggression. And when we started the project, basically there were two different paradigms. One of them, the Taylor aggression paradigm. I don't know if any of you know it, but it's a quite nasty paradigm where you actually you'll get hurt by electrical shocks, etc. So it wasn't really what we wanted to do. It would also be a little complicated to do in the scanner. So we went for the point subtraction aggression paradigm, which is basically a game. So you tell the participants that they are about to play against another player, and that player is situated in a different scanner. And so we have this whole act about, you know, when we're setting up, we have to wait for the other group. And what you can do is you can earn points by pushing a button, and you earn, once you have earned 100 points after pushing the button a certain number of times, then that adds, and you get paid for that. So you get, you get paid for the points you get by the end of the game. However, from time to time, the other person in the other room is going to steal your points, which is quite annoying. So, then you can choose to react different ways. Either you can just continue to earn, which is really the only reasonable thing to do, or you can steal the points back. However, you don't get to keep the points. So it really doesn't give you any benefit to steal points except for the satisfaction of retaliation. And you can also choose to protect your points. So there are these different ways that people can react. Yeah? Yeah.
How do we make sure that the person, when on the scanner, are angry or not, or how long passes from?
Yeah. So the nice thing about the point subtraction test is that you can actually see how they respond. So if they have a number of aggressive acts, that is stealing points that they don't get to keep, which is just a waste of time, but it gives them satisfaction. So that is one measure of, it's a behavioral measure of how angry they feel and how provoked they are. And that's how we measure the actual paradigm. That's their response within the scanner. So here you see the setup. You have these different choices. You can select different buttons. And then from time to time, there's somebody stealing your points. Very annoying, because it's actually a pretty boring game. And so it's very annoying. You earn all this money and for no good. So you have a question here.
Do you just check whether they believe that there is no?
Yeah, we did. And we asked them afterwards. We asked them different questions. So we did hide this question so it wasn't too obvious. And there were actually, I think, a couple of people who said, well, I had the sense that this was not another person, but it was a computer. And we did actually discuss whether, the plan was to discard those subjects, and we did. But actually, when we included those subjects who did not believe in the deception, they fitted nicely in. So I think that it doesn't really matter. And if you play a game and you feel that you're not doing well, you actually get very annoyed, even if it's not a person. So I think it actually, maybe it doesn't matter too much whether they believe in the deception or whether it's a computer. I mean, you can also get mad with your computer when it doesn't work, can't you? So that's, I think it doesn't matter that much. So they're provoked, and then they can do this aggressive behavior. And sure enough, what we found was that the violent offenders much more frequently chose the aggressive response compared to the healthy controls. But again, you can see that there is quite a spectrum, and some of the healthy controls actually also chose to choose the aggressive act. And question? More questions?
So in terms of the violent offender, couldn't it also be that they were not more aggressive to the general population, to just have difficulty inhibit their aggression because they had subfrontal?
Well, that's also what we're looking at, because we will also look at the connectivity. So that's an interesting question. There's another. Yeah.
So the self-report personality test, if the violent offenders were they aware, did they report that they thought it was aggression, or they thought it was?
Oh yeah, so the question is, when they filled in the questionnaires, the self-report questionnaires, were they aware that they were recruited because this was a study of aggression? And I would say we tried to be a little vague about it, because I'm sure they wanted to make a good impression on us, and particularly on the project leaders. They wanted to be on good relationship with the investigators. So we were not too open about it, but I think they must have known somehow that this was related to their behavior. So there was another question here.
Is this a measure of aggression, or is it a measure of how much people care about social norms, if that makes sense? So they might, for example, the control subjects might feel a bit bad about stealing from somebody else. They do because somebody else is there. Whereas the aggressive offenders might actually just feel they don't really care about somebody else.
Yeah, so what you are touching upon here is also another interesting question, which is, so why are the violent offenders? Why are they in prison if they have an aggression level that is comparable to people outside? And I think it has to do with social norms, as you say. It's what is acceptable, and that's so. It's an interesting question.
Isn't it's better that when the patient or when the person or in the scanner use
show that some movies or pictures that make them angry, and after that,
Yeah.
that's the aggression.
So the proposal here in the front is, could we have thought of another paradigm in the scanner that would make people angry, such as a video or a movie clip or something like that? I think that, you know, in order to get angry in the aggressive, impulsive way, it needs to be personal, and that's why we wanted them to believe that somebody was trying to harm them deliberately. If you're watching a movie, it's not that personal. I mean, you could watch somebody, you know, beating up each other, or you could do something else. But I think it had to be something that they were engaging in and where they felt that they had been provoked personally. And we also did, I'm not going to show the data, we also did some emotional faces, and we have some different outcomes for that. But yeah, more questions?
Was the game like only losable? Like, can they win also win money?
They all won money. They all won money, but they didn't lose. We didn't take money away from them. I didn't want to do that. Yeah.
Was it not possible to make them think they were rather shocking someone else, or something that was actually more harm rather than theft?
What are you thinking of? You're thinking of some more harmful things than playing a game?
Yeah, rather than theft, just be like, okay, if you press this button, the other person can get a shock, maybe they get a shock and so on, or is this sort of...
Yeah, that was the Taylor paradigm, which works outside the laboratory. I mean, there are two things to be said about it. I think, first of all, I didn't really want to risk that we had some episodes in the scanner, obviously, for obvious reasons. And secondly, as you will see, it actually worked. I mean, it doesn't have to be... I think the lesson to learn is that some people respond to acts that some of us would not consider as provocations, but they perceive things they encounter more easily as provoking, and then they cannot inhibit their response. So that's what we see. You had a question too?
Were any of the healthy controls as aggressive as like the blind offenders?
We'll get to that point. I can show you the distribution when we get to the aggression measures. Okay, so what we found was that, because I was curious to see, so is it in the act of retaliation that they actually have a reward? And interestingly, when we did the first study only in healthy controls and we established the paradigm, what we saw was actually that during the retaliation, that is when they were stealing back points, they felt apparently some kind of pleasure in doing that, because we did see activation in ventral striatum. However, we didn't really see this in this sample, so where we really saw a striking difference was during the provocations. So I think it's interesting to distinguish between to what extent do their acts satisfy their needs, and I think to some extent it probably does. And of course you can never mimic a real world situation in a scanner, but I think it was interesting still that the provocations was really where we found some striking differences. And what we could see on a group level basis was that when the violent offenders were provoked, they had higher amygdala reactivity compared to the non-violent controls. And we also saw some striatal activation that was higher in the violent compared to the healthy controls. So there were distinct group differences at this level in two regions that we had hypothesized would be involved. And what is quite reassuring is that when you do some of these paradigms, you really like to see that there is some kind of association between how they behave within the scanner and the reactivity. And here you can see that the green ones are the violent offenders, and the blue ones are the healthy controls. And you can see that there was also, when you looked at it, not on a group basis, but on a spectrum level, there was actually an association between their number of aggressive responses within the scanner and the amygdala reactivity, which to me was quite reassuring. And that was also the case for the striatal reactivity. meaning that it does seem to have a meaning for what we find here. So, okay, so when we looked at the connectivity, again, we're interested in looking at how well does the prefrontal cortex manage to interact with the amygdala. And what we could see is that the healthy controls had a higher connectivity between the prefrontal cortex and amygdala. And that makes sense because you are provoked, but your prefrontal cortex tells you, well, calm down, there's no need to act on this. So the connectivity between the two regions was better, if you will, in nonviolent offenders, and the same applied to the prefrontal cortex and striatal. So what did we see when we looked at the PET scanner then? And maybe this is a good time just to show you a little bit. What does it? Oh, there's a question. Let's take that.
So the statement was when provoked, healthy controls had higher connectivity.
Yeah. Yeah.
So that means that steady state, there was no difference between the healthy controls and others.
And the connectivity, you mean at baseline, resting state? Resting state. We have not looked at resting state yet, actually. So we just completed the study, 1.5 years ago. So we've been busy completing all these things. But it's a good question, and of course we'll look at the resting state connectivity likewise. So, more questions?
So, did you also test if they had any pleasure just from stealing from the other person, just to see if it was...
You mean by asking them?
No, like during your MRI, just to have like one option that just said steal something from the other person, just like, not related to any sort of provocation, but just to see if they actually had some sort of pleasure out of that, if it was connected with the pleasure of stealing,
yeah,
or if it was connected with like retaliation.
Yeah. So we actually did see a relationship between how often you stole, and in the moment of stealing, we saw a ventral striatal activation in our first population, which consisted of healthy people only. But surprisingly, we did not, replicate this in this sample. And this is sometimes what happens. And particularly, I would, of course, have thought that the aggressive people, they would have more pleasure out of what they did. But that didn't really show up in our data. So, yeah.
So have you checked also the function connectivity with the divide of investigation
So have we checked the functional connectivity in healthy versus violent?
This is the healthy one. Also, have you done similar experiments?
Oh, okay. Yeah. So, but that's what it means when. So, sorry, but this is a group comparison. You could also have said here that when provoked, the aggressive impulsive individuals had lower connectivity. So it is a group difference where you contrast the two groups. So, yeah. Is that clear? Yeah.
Again, where present are in the scanner or not angry. Okay, so I think this result is related to the situation of coping by the aggression that is different in different people.
Yeah. So exactly. So what you said here was that. I'm not sure if they're not angry when they're provoked. I mean, actually, we had a question afterwards, and we told them whether they were annoyed. We didn't ask if they were angry, but we asked them if they were annoyed. And they were annoyed. I mean, also the healthy controls were annoyed, because it is annoying to be provoked and that somebody's stealing from you. But this connectivity, you could say, is also something about how you cope with the provocation. And that's exactly how I think we can interpret this. Yeah.
I'm sorry, I have one question. Right, higher connectivity with I assume actually it's a correlation. So there should be a higher correlation. But when you give the cluster size, you also say it's more spread or there are more voxels. Is it both important?
What do you mean more? Okay. Yeah, okay. So I think, no, yeah, so what you're saying is that I have a statement here and then I have a cluster size down here. So this is just to tell you how large is the cluster. So the p-value is also taking into account a threshold of the cluster size. So that's why it looks like that. Okay, another question?
Okay, so you also said that amygdala activity was higher in
Offenders.
in the offenders. So
Yeah.
did you control for that when you did the connectivity?
I don't think so. Should we?
It might have an effect that the activity is different than.
Yeah, but wouldn't you think it would go the other way then? That if you had higher amygdala reactivity, you would have more connectivity? Then, because it's kind of counterintuitive that it would go this direction. So, I mean, maybe if you accounted for the higher amygdala reactivity during the provocation, you would actually have seen an even larger difference, group difference. I don't think we did that, but I don't remember on the top.
I have a question for you. Someone asked about resting state. You haven't done it on this occasion, but supposing you had.
Yes.
And you found similarly the difference in connectivity, obviously the absence of provocation.
Yes.
How would you interpret that, or how would you interpret no effect?
Yeah. So how would we interpret if they actually did have less connectivity in the resting state? I think that resting state connectivity is a very different condition than in a provoked situation. But having said that, your question is really, could it potentially have an impact on how we interpret this so that it wouldn't have much to do with the provocations? I don't know. I think that our resting state is a quite different situation, because what you often do in a resting state, it has been shown that the resting state default mode network is very much related to what you do while you're in the scanner. So you're mostly thinking about yourself and autobiographical things. I did this yesterday, I'm doing this tomorrow, and this afternoon I need to do this and that. So it's very much centered about yourself. And I'm not too sure that we would have a very strong connectivity between the prefrontal cortex and the amygdala in that situation, actually. Yeah.
How do we measure behaviorally the lack of inhibition control when they can't do anything, like they're not actively not inhibiting, or they're just feeling that, oh, they're annoying, but they're not actually actively.
But they are doing an active act because they are deciding that they want to do a retaliation and they want to punish the other player by stealing points from that player. So they are actually doing an active thing in the scanner.
Okay, so it was different from stealing points versus just gaining money, like gaining points?
So most of the time they'll spend gaining money. And then, occasionally they get provoked, and sometimes they respond immediately, and sometimes they just feel, oh, I think I'm going to steal that annoying bastard in the other scanner. I'm going to steal some money from him. So it's both related to provocations, but it's also unrelated.
So isn't it better that subjects compare with themselves before aggression and after aggression, because the capacity of their brain or connections in subjects' brain are different?
So your question is whether you could compare connectivity between prefrontal cortex and amygdala in a more neutral setting, I guess, because then you wanted, so the idea is that you could compare the subjects to themselves. And that comes back to what I mentioned before, in the beginning, some years back, when we started applying fMRI and combining it with a PET. I was also under the impression that it would be better to use people as their own controls, but I've actually, I'm not convinced anymore. I think that the inter-individual variability is not much different from the intra-individual variability, which is kind of surprising because you would think that people would be responding more or less in the same way. Andrea?
I have two questions. The first one is, did you test IQ?
We did test IQ, and that's a good question. IQ was slightly lower in the impulsive, aggressive individuals, which comes back to the comment I made before that often it's actually hard to find people who are not well educated with a high IQ. But it wasn't all that bad, and we did, I don't think it's stated here, but we did actually add IQ as a covariate, just to make sure.
And the second question was, did you see a difference pattern in the pleasure of getting money? Without the aggressive stealing, but just like the baseline?
We did look at the wins as well when they won the money, because every time they passed a certain threshold, they would get points and they would win money. But that we didn't see any difference in terms of the pleasure and the rewarding experience of gaining money. Tony?
I might have missed this, but I know that functional connectivity goes up whenever two areas are co-activated. Does functional connectivity also increase when one area suppresses another area?
Well, that's a good question. Does functional connectivity go up when one area suppresses another area? And that has to do with directionality, because with this analysis, we're not really sure what comes first. So it's a good question. I don't know. But this is the best you can get it, because probably what you would need to do, I guess, would be to do a higher time resolution study where you would looking at the sequence of doing more like a dynamic modeling, perhaps.
Because there's a literature showing that especially medial prefrontal cortical areas can suppress amygdala responses to stressors. And there's also functional imaging literature showing that when people actively suppress an emotional response, there's an activation that occurs in the frontal cortex and failure of activation of the amygdala.
Yeah.
Suggesting that when they don't respond to this provocation, it might be a frontal cortex suppression.
Right. But wouldn't you expect that in the case where you have a suppression of the amygdala, that the signal would perhaps go down in amplitude, but you would still have coherent oscillations? So, I mean, that's really what you're looking at here, is not the signal size, but it's more how coherent it is in its shape. So.
Does it show up when they're anti-correlated?
Excuse me?
Does it show up when they're anti-correlated? So when activity goes down in one area and goes up in the other?
No, this analysis just shows you something about how coherent these signals are in amygdala versus prefrontal cortex. That's what this analysis tells you, and that's all. Okay, lots of questions. Okay, so let's just talk about, some of you know something about PET, some don't. I just want to briefly tell you what it involves. First, we have the physicists getting up at 4 a.m. They turn on the cyclotron and they produce the positrons. Then you have a radiochemist, and once the production is made, he has approximately 1 hour to do the carbon-11 labeled compound, because the half-life of these compounds is 20 minutes. So if he spends more than 1 hour, then forget about it. You're not going to do a study. You have to wait. So they need to be clever and very patient. So they have a very patient personality in general, very pleasant people, I'd say. Then you do the quality control and you inject it into the subject. And here you can see the armed guard sitting next to the PET scanner while Sophie is working here. We need a number of people engaged in this. We have there's also, often we need to do metabolite analysis, because the moment that you inject the radio tracer, it starts to metabolize. And in order to account for that, you need to have a technician doing HPLC, so you can determine the parent compound for the input function. Or perhaps if I had time, I'll get a little bit more into that. You need to have a person to acquire and reconstruct the images, so you get a 3D image of the entire brain. And then once that is done, then you do the PET and the MR. You also do an MR image of the individual. And then you do a co-registration of the PET to the individual MRI. And once you've done that, then you can put in automatically, you can add an atlas, you can extract brain regions, and you can then get some time activity curves. So this is time and this is radioactivity in different brain regions. And then you need to do the modeling to get a quantitative measure of the binding potential, which is what you finally end up with. And then we're back to the first images I showed in last session, that now you can actually, if you want to, you can convert that into protein densities, but mostly we just relate the binding potential across group, kind of assuming that, that the affinity to the radioligand of the receptor is more or less the same. Okay, so what we found was that there was a group-dependent association between striatal 5-HT1B receptor binding and trait. So here you can see the trait. So now we are back to the questionnaires where you were filling in questionnaires, and this is a combined measure of the trait anger. And what we saw was that in the healthy controls who did distribute, you can see the distribution of traits here that did distribute, but they were not massively aggressive. You can easily tell that some of the violent offenders were definitely more aggressive, but you also had some offenders that seemed to fall in the healthy range down here. So they came with different traits, not surprisingly. And what we saw here was that in the violent offenders only we could see an association between the striatal 5-HT1B receptor binding and this trait, but not in the healthy controls. So it seemed like the violent offenders had higher 5-HT1B binding in striatum than did the healthy controls in general. And when we looked again at the striatal 5-HT1B receptor binding, we could see that again, so this is the same figure I showed you on the last slide, that trait anger was associated to the binding potential in striatum, but also trait psychopathy had some relationship to this, and also PCL-R score, which is another measure of aggressiveness, was associated. So you have all these different measures of traits, and of course, this is one of the complications you have when you do in vivo imaging. Now you have a number of regions, you have a number of traits perhaps, so you're bound to get into problems with multiple comparisons once you start to invest. I mean, you most likely, if you do 20 comparisons, you are going to find a significant result in one of these data sets. So I'll get back to later how you can try to cope with this. And one of the ways to cope with that is to generate a so-called latent variable model, where you try to capture features of aggression from the different scales. And this is something that we are just doing, we're doing very generally with our new psychological test scores, because often you do these tests and you tend to have overlapping tests, and maybe they capture elements of different things, but by defining a model that can sort of home in on one latent variable, then you get rid of, first of all, you hopefully get a more precise measure of what you are after. Secondly, you get rid of the multiple comparisons. But anyway, we could see that it was actually seemed to be relevant for a number of traits in these people. Okay, so just to summarize here the outcome measures we had here, that serotonin 1B receptor binding across brain regions relevant for aggression named here. So these were the aggression-related circuits that we were, or brain regions that we were interested in examining. We could also see that the reactivity to provocations was particularly in amygdala and striatum with our fMRI measures. And then we had the trait anger, which showed that the violent offenders had a general propensity to experience angry feelings. So now the question is, how can we combine these different measures and get some better idea about what is going on? And this is the latent variable model I was talking about. One thing I have not mentioned is that in general, when you do in vivo imaging data on receptors, what you find is that if one person is high in, say, anterior cingulate 1B, that person also tends to be high in all other brain regions. And the way that this can be interpreted is either because you have some methodological issue, could be the affinity is slightly different, could be that the free fraction is slightly different, but it could actually also be that you have a denominator in the raphe where the projections are influencing the actual receptor levels in those regions. This is something we often see. Over here we have the amygdala reactivity and the striatal reactivity and trait anger. And when we put together a latent variable model where we say that the 5-HT1B is a common denominator, you can see that these different regions, they load differently onto the latent variable 5-HT1B with some of these beta values. So what you do is you choose a reference region, and in this case we chose the anterior cingulate to have the beta value of 1, and then the strength with which these different regions are associated to our latent variable is given by the beta values, and they are all about between 0.7 and 0.9, so they load quite well onto this latent variable. So how does that relate to our amygdala reactivity and striatal reactivity? And it turns out that we can explain a fairly large fraction of the amygdala reactivity through the latent variable here for both the amygdala and the striatal reactivity. And now the final question is, how does trait anger map onto this? Does it have an independent effect on striatal reactivity and the latent variable, or does this act through the 5-HT1B? And in fact, what we see with this model is that the trait anger seems to operate through the latent variable 5-HT1B, so that the reactivity that we see with the functional imaging is mediated or explained by the 5-HT1B receptor. So I know this is a little complex model, but I think it's very informative for us to think about how we can pull together these behavioral and trait and receptor features in a consistent model. Andrea?
I just did a quick search on the receptor type, and it has been linked to smoking. So did you count that in your analysis? When was the last time the individual smoked, or if they were smokers?
Yeah, so the question is if 5-HT1B is affected by smoking, and you could think of it in two different ways, I guess, that if you have a certain 5-HT1B phenotype, you're more likely to smoke, or you could also think of it the way that maybe smoking alters your 5-HT1B receptors. We did look into that too, and we did add that as a covariate and did take that into account because that's something that can, that's important too,
Do I suspect correctly that you interpret this as reflecting mostly postsynaptic compensatory upregulation?
So that's a very good question. So is this latent variable, is that due to presynaptic 5-HT1B or to postsynaptic 5-HT1B? And I would love to give you the answer, but the truth is nobody really knows. I haven't been able to find any data that tells me how much of the signal that I see of the total is actually situated presynaptically versus postsynaptically. It differs a little bit. Some people tend to think that it's more presynaptic, others tend to think it's more postsynaptic. I think it would definitely be really interesting to know, also to get a better handle on how to interpret these things. I mean, it would make sense if it was more the presynaptic measures.
If you had also imaged the transporter, you might have gotten some idea which direction things are going.
Yes, except that you cannot really use the transporter to tell you what fraction of 5:1b is imaged.
Yes, but you could get a sense which direction the system is going, right? Because if it's free versus postsynaptic, that would give rise to a very conflicting interpretation.
Yeah. So we are actually starting to do some of that, not in this population, but in a different population that had both serotonin transporter scans and 5-HT1B scans. And interestingly, there seems to be, I mean, this is preliminary data, but it seems that there is an association between the two of them, which I don't know how you would interpret, but to me that suggests that there might be more of a presynaptic component in what we measure here with these imaging methods. Yes?
These results show that if you block this receptor pharmacologically, if people don't get angry.
Well, that's, of course, that would be the next step. So the question is, if you pharmacologically intervene with the 5-HT1B binding, could you alter their aggressiveness like you can do in the rodents? And the short answer is that nobody did that ever. But I think studies like this is what could be, very interesting to move forward with and look for pharmacological targets, because it's a very devastating condition, not only for, of course, the people who are offended, the people who are suffering from the violence, but also for the offenders themselves and for society, because it's really expensive people to have, locked up. Yes?
Was there a big difference in the type of crimes they committed in these offenders? I mean, like for something like physical violence versus like deception or psychological,
harassments or, yes. So, no, I think the types of crimes they had committed differed in severity from a manslaughter to, and also repeated manslaughter to, they were raping and they were just, beating up people. But, if you beat up people severely, then sometimes it's more a question of, how fortunate the victims are in terms of surviving. So, I mean, the distinction is, so they were all really bad guys, basically. Yeah.
Did you take if specific activity had any influence on your binding potential?
Specific activity? Oh, you mean in terms of, yeah. So, so I mentioned before that that you need to be at tracer doses. And oftentimes, when you have a fairly new radioligand, you need to examine that you are truly at tracer doses. And what we did to make sure that, because sometimes the radiochemist takes a little longer to make the production, and then the number of radiolabeled molecule out of the total molecules is smaller than if he's really quick. So you should think of it this way, that perhaps when you inject a radioactive dose, then it's maybe 1 out of 1,000 molecules that's labeled. So it's not all the molecules that are radiolabeled. And as they start to decay, then there'll be less and less specific radioactivity because you will get rid of the radioactively labeled molecules, obviously. So if you're in a non-tracer condition, which is the suggestion here, do we control for that? So what you can do is you can look at injected mass, and if you inject a cold mass that is high in one group and higher in one group than in another group, that could theoretically drive a difference. And we were fortunate here that they all got a very high specific radioactivity. We also added that as a covariate, and we didn't find any significant influence of the injected mass. So you need to make sure that you have tracer doses. So that is part of the routine validation you do of any study to make sure that, and that is what is becoming challenging if you want to do PET studies in animals, because here you have a small animal and you need to have enough radioactivity for you to pick up a signal. And then it can sometimes get a bit complicated because you inject too much of a mass dose and then you start to block the binding, so you find lower binding potentials in those cases.
Gitte, can I ask you, I think this is a really interesting case. If you were able to do an animal study with PET, I'm sure you could do all
yes.
sorts, what could you add to this analysis, you know, if you'd like to know, with an animal PET study, for example?
I think what we would like to do in an animal PET study is to do a pharmacological intervention on the 5-HT1B, to do maybe, I don't know, I think knockouts are a little, you know, I'm not too trusting because they can adapt and things can happen. But to make measures of aggression levels in rodents, so there are some optogenetic methods that you can use to induce aggression, and you could potentially see if that would, if you were able by changing this latent variable either pharmacologically or genetically to turn it on and off, whether you could actually tell, because there's also going to be some inter-individual animal variation, hoping that that would be large enough for you to actually assess that. So that could be an interesting experiment to do. Yes?
When you do alpha males and submissive males, and then compare them, if you take like very old male mice and put them together, they'll fight and there'll be an alpha male, and then compare like the genetic expression.
Yeah, but your question is kind of assuming that alpha males are more impulsive aggressive, and I'm not quite sure that that's the same feature that we're examining. It's more about being able to inhibit an aggressive response. Yeah.
Did you try to look at if you don't split the groups between like the offenders and the non-offenders, but between like high aggressiveness and low, high aggressivity and low aggressivity, because being an offender could just be like being in a bad environment, and
exactly.
not being an offender could be being in a good, or
yes.
Yes. Being able to like channel your like
yes.
force or whatever. I mean.
Yes. So in this model, what we did was to take everyone. So we took all the subjects, the healthy controls and the violent offenders. So we are looking at it as a spectrum. You're proposing to split the group in a different way so that you just take all those with high trait measures and compare to those with low trait measures. And of course you can do that, but I think that you will be missing out some very important data that come out of the variation that you look at by looking at correlations rather than looking at group differences.
Yeah, but then it would still be like on a spectrum, like yeah, okay, but that's actually what you do.
Yeah, that is what we do.
Oh, yeah, okay, sorry.
Yeah. Yeah.
How old were your subjects?
They were, how old were our subjects? They were, if I remember correctly, something in the range of between 22 and I think the oldest was 58. And we did take age into account as well. Yeah, because there is a quite profound age decline, so you need to take that into account as well.
And these changes are a kind of scientific plasticity and what routine. Do you think, is it reversible?
So to what extent is 5-HT1B binding reversible? I don't know, because to be honest, I don't know the answer, but I know that 5-HT1B is going down with age, and we certainly see that a lot of violent offenders, they tend to become more calm as they get older. So, I mean, it's consistent with this thing that 5-HT1B could potentially be involved, because you actually do get less signaling through 5-HT1B as you age. And as we know, people also tend to get, calmer with age. So, yeah. Another question?
Were all the aggressions rewarded in this test? So if they always wanted to steal, they would always get something back? Or was there something?
No, they didn't get to keep the points. So that was the point. So that was the whole point of the game, that, and interestingly, some of the offenders, they, when they heard about the rules, they said, hey, that's not fair, that's unfair, I don't want to, this is an unfair game. So, well, that's how the game is. And the other person plays by other rules, that's what we told them. But they were pretty annoyed. Yes?
Sorry, I might have missed this. Is there a relationship between trait macular and how much do you steal points?
I don't remember if we did that specific analysis, but if you think about the first slide I showed with the data, you could definitely tell that the aggressive impulsive individuals, they punished more often than the healthy controls. But I'm not sure we did that exact correlation behaviorally. What was most important to us was to know if we had a behavioral response that matched the fMRI response. That was the key thing about their responses. But there was a group difference in how they responded, as we would expect.
Mine is not really a scientific question, it's more like bioethical.
Yeah.
I'm trying to understand what are the possible outcome of research like this. So do you think that we have like a particular involvement of these receptors or with some particular area of the brain, this will help us to consider maybe aggressivity more like a psychological disorder instead of, and so maybe these aggressors, instead of going to jail, they will go to a hospital and have a cure.
Yeah, so that's of course an interesting point. So there was a documentary made actually on exactly this project where there was a journalist who found a highly aggressive offender who was 28 years old. He had 7 times been convicted for violent acts, and they wanted us to do an fMRI study on this person. And the whole idea was that, you know, to start the entire discussion about to what extent can features like the ones that you can identify in this type of projects, how can they be relevant for how you convict these people? I mean, we all know that if you're psychotic, when you do the act of crime, you can be convicted to be treated, and you are put in a hospital, and you are not, you know, put to jail. And then the question is, if we were able to come up with a good pharmacological target, and if we were able to treat these people, could you then potentially think about how would you then change the way that you convict people? And I'd say that possibly. I mean, it depends. I think that morally these people are still responsible for what they do, regardless of how their brains look. And actually, particularly in the US, you're starting to see some trials where they bring in additional evidence, like could be genetic polymorphisms that were particularly associated with violent behavior. It could be if they had a particular bad upbringing, which is also something that could add. So it's not my impression so far that it has had a huge impact, but in some cases they do try to make the case that they should have a more lenient conviction.
Because of that, I said that I think it's better than the subjects compare with themselves. Because the life experience.
Yeah, if you could do an intervention and then you could see if they became less aggressive, that would be the perfect experiment to do. But we need a good drug. Okay. Actually, I had a whole other presentation, but that's fine. Very good questions. I think it's time for us to do a Kahoot.
So that's it. That's it. So,